أهلا وسهلا. Lecture of Clinical and Chemical Pathology, Cairo University. تفضلي.
Good afternoon, dear ladies and gentlemen. I'm really grateful to be here today among my big family. And let's start. I'll be talking about artificial intelligence. Bismillah ar-Rahman ar-Rahim. On the 30th of November 2023, a magazine called Plus 972 magazine released an article. But before going through the title of the article, let me tell you some information about this magazine. This magazine was established in August 2010 by a group of 4 Israeli writers from Tel Aviv. They wanted to express a new voice which would take part in the so-called international debate regarding Israel and Palestine. And the name 972 is the international dialing code that is shared by Palestine and the occupying Israel. The title of the article said, "A mass assassination factory, مصنع اغتيالات جماعية, inside Israel's calculated bombing of Gaza, permissive airstrikes on non-military targets and the use of an artificial intelligence system have enabled the Israeli army to carry out its deadliest war on Gaza." Ladies and gentlemen, after that article and since that date, almost every news channel, every newspaper, every news magazine talked about that artificial intelligence program. And they named their program the gospel. Do you know what's meant by the gospel? It is el-Ingil. And it's a word that is derived from Old English that means the good news, al-Bishara. On the 1st of December, The Guardian said the gospel, how Israel uses AI to select bombing targets in Gaza. They said that it has significantly accelerated a lethal production line of targets that officials have compared to a factory. سموه مصنع وبيعمل خط إنتاج مميت من التارجتس. بيجيب الانفورميشن بتاعته منين؟ من drone footage, intercepted communications, surveillance data, and information drawn from monitoring the movements of behavior patterns of individuals and large pools. This is the dark side of artificial intelligence. AI could advance in a way that surpass our wildest imaginations, and it could radically change our everyday lives much sooner than you think. And from here, the ILOs of my session, استاذن هو حاجة حصلت هنا؟ كنا I'm sorry. So the ILOs of my session, we will be talking about definitions and levels of artificial intelligence, machine learning and machine learning methods, application of AI in various disciplines of hematological diagnostics, future considerations and challenges ahead, conclusion and take-home message. When it comes to technologies we use to make medical decisions, we need to understand the core of the machine, or at least the physical and biological explanations behind it. So what is artificial intelligence? In its simplest definition, it is intelligence demonstrated by machines, also described as machines that mimic cognitive functions that humans associate with human mind, such as learning and problem solving. So here I will be going through levels of artificial intelligence. The first level is artificial narrow intelligence, which is an algorithm that can excel at a precisely defined single task. هو احنا عارفين هو عايز يعمل ايه؟ هيعمل حاجة واحدة and it recognizes abilities in huge data sets. The second level is artificial general intelligence, and this is human level AI. It can reason, argue, memorize, and solve issues like you do. One day could have a human being's comprehensive and total cognitive capacity. The third level is artificial superintelligence, where theoretically it has the humanity's combined cognitive capacity or even more. Humanity obviously would not be able to grasp its knowledge and understand its reasoning. These are the three levels of artificial intelligence. لو طبقناهم, the artificial narrow intelligence, it's like the GPS. هو عنده single task عارف يعملها, ولكن عنده a lot of data وبيطلع في الآخر single task بيوري الطريق. This next is the artificial general intelligence, which functions like a human being, and the artificial superintelligence زي ما قلت ده في the intelligence of the whole humanity. الدايرة الخضراء هي the ideal AI. How does artificial intelligence work? It works through machine learning. What is machine learning? It consists of computer algorithms capable of learning tasks from data without being explicitly programmed. These algorithms do not operate on pre-programmed rules, but are allowed to decide on rules on themselves by finding patterns in data. If you feed the algorithm with enough data of good quality, machine learning allows them to create strategies for excelling at particular task. لو احنا عايزين نعمل program to spot cats in a picture. البروجرام مش عارف يعني ايه cat. أنا لو جيت قلت له the cat has 2 ears, 2 eyes, 4 legs, هو مش هيفهم, because what is an ear for a program that only sees pixels on a photo? Instead, machine learning, we can introduce a lot of pictures of good quality containing cats in them. لوحده هيعرف بعد كده to spot the cat in the picture. Methods of machine learning. Supervised learning, used when we can precisely define the task we want the algorithm to learn based on data that we already have. It's like learning with a teacher. The second is unsupervised learning. It is like learning without a teacher. Using unlabeled data, it tries to explore data to find patterns and features. Reinforcement learning allows the algorithm to learn how to complete the tasks with a sequence of decisions by itself, without being told how to do it. The teacher is only able to give feedback after a series of actions, not for each item as it does with supervised learning. لو طبقنا الكلام ده في الهيماتولوجي the supervised learning هندخل label data هيطلع لنا predicted outcome. لو حطيناه على المورفولوجي بتاعنا using diagnosed pathologic slides the supervised learning can be used to create a model that is able to classify slides with the disease. The unsupervised learning بتدخل unlabeled data بيقدر بعد كده to identify patterns أو يعمل groupings. Example, identify clusters of similar gene expression profiles among B-cell lymphoma samples. ولو طبقناه في المورفولوجي بيقدر by using undiagnosed pathologic slides it can be used to identify slides with similar patterns. آخر حاجة reinforcement learning بندخل data هو بيطلع the predicted best action ولكن عندنا فرصة to give feedback to improve. ولو حطينا ده على المورفولوجي using pathologic slides with only some with final diagnosis it can be used to create a model that is able to classify slides with the disease. أشهر example to the reinforcement learning هو ألفا زيرو. ده برنامج بيلعب أي two player game بيتعلم إن هو يكسب إزاي عن طريق إن هو playing millions of games against itself. Deep learning. Deep learning is a subfield of machine learning which uses deep neural networks of multiple layers of artificial neurons that learn their own level of representation of the data by repeatedly updating the parameters of the neurons in a process called training. The neural network is a computer system modeled on the human brain معمول بطريقة إن هو multiple layers وكل layer بتنقل الداتا لللاير اللي بعدها. Now application of AI-based methods in various disciplines of hematological diagnostics. Number 1, cytomorphology and histology and immune histology. Number 2, multi-parameter flow cytometer. 3, chromosome banding analysis. 4, molecular genetics. First, cytomorphology and histology. قبل ما نعمل model training محتاجين شوية data pre-processing. Number 1, image digitalization. It's the process of converting histology glass slides to digital images. Number 2, whole slide imaging. It's a technology to digitally scan and archive whole slides in high resolution. During scanning, images are taken for each view. Simultaneously, they will be stitched together by software to create a single digital image file. عندنا بعد كده image segmentation. It's used to partition an image into multiple parts or regions, often based on the characteristics of the pixels in the image. بالشكل ده لو طبقناه في البون مارو عندنا هيعمل image segmentation, object identification, feature extraction وبعدين classification of the different cell types. Here is an example of the output of a neural network capable of segmenting 6 different cell types within the bone marrow, giving each cell a color, and it could also give estimates for the cellularity. Now model training هنختار أنهي machine learning method for the training. The choice of the category of machine learning to apply to a specific task is a non-trivial decision. There are often different solutions for the same task and frequently different algorithms are combined to perform a task. هنا example number 1, supervised learning عمل extensive fine grain annotations لكل mega karyocyte علم عليها فبالتالي the machine learning model could be able to detect all the mega karyocytes. Semi-supervised learning هنا علم بس على the hematopoietic areas فبالتالي the algorithm could detect the hematopoietic areas. weekly supervised learning هو اداله MDS slides و slides with reactive dysplasia. في الآخر هو قدر إن هو يحدد the reactive slides. The unsupervised learning هو اداله مجموعة من السيلز من غير ما يعمل annotation لأي سيل. في الآخر قدر إن هو يعمل clustering of the cells on morphology. The most method used is the deep learning because it has high accuracy and ease of implementation. Applications of AI to bone marrow histology هتبقى identifying of bone marrow cells and structures, recognizing disease-specific algorithms, and phenotype-genotype correlation. اتحط كتير من البرامج اللي هو قدروا يعملوا identification of the bone marrow cells and structures. دي example لهول slide imaging. زي ما قلنا بيصور كل field لوحده ويجمعهم كلهم وبعد كده يقدر يعمل region classification, then cell detection and cell classification. The evaluation of the bone marrow biopsy is much more complex due to tissue architecture and variation in object shape due to cutting effects. وده هنا برنامج قدر يحدد the intrasinusoidal infiltration. تاني حاجة recognizing the disease بعد ما عملنا identification to the cells. ده بيتعمل عن طريق application of deep learning algorithms. Disease-specific algorithms, one of the most attractive study subjects in bone marrow biopsy is the MPN. كان في study قدرت إن هي تعمل library for the megakaryocytes عن طريق applying deep learning methods. لو جينا هنا أخذنا example للموديل ممكن يتعمل ازاي. machine learning based image segmentation هيقدر يحدد single cell images. يدخل بعد كده على deep learning based cell type classification ويعمل cell differentiation results. يجي هنا دور الexpert يا إما هيعمل diagnosis يا إما هيعمل correction of the misclassification to continuously improve the model. أو ممكن يمشي عن طريق deep learning based model يعمل extraction of morphologic features associated with genetic aberrations ويعمل يدخل بعد كده على expert evaluation. تاني حاجة multi parameter flow cytometer. Still عندنا data pre-processing. The raw expression values are transformed into images. There are two typical sensors اللي ممكن يستخدموا هنا اللي احنا عارفينه photo multiplier tubes ودي single pixelated devices هتتحول أو هتتغير ب multi-pixelated imaging devices camera based. Two main types of computational methods for clinical flow cytometry data analysis can be discriminated. حاجة اسمها dimensionality reduction. It allows for two-dimensional visualization of the multi-dimensional flow cytometry data and clustering. الclustering هيجمع كل الcells with the same expression of markers مع بعض. Chromosome banding analysis. Karyotyping is a very time-consuming and complex task, and a high degree of automation is desirable. The challenges for automated karyotyping are multifarious and not yet fully resolved. هنا برضو بنفس الطريقة machine learning based image segmentation plus geometric transformation هيعمل chromosome classification ويجي دور expert evaluation يا إما يحدد يا إما يطلع the diagnosis and prognosis ويرجع يعمل correction of misclassifications to improve the model. Molecular genetics, genes, panels, genomes, and transcriptomes. while the other fields integrate AI technologies to mimic human intelligence and to reproduce the knowledge of experienced diagnosticians, clinical molecular genetics aims to implement machine learning-based methods to perform tasks that are impractical for humans to do. Basically, every step in clinical genomic analysis could benefit from the integration of machine learning, but not limited to recognition of DNA sequence patterns, variant calling, and variant effect prediction and classification. Future considerations and major challenges ahead. Input data challenge. The performance of the algorithms largely depends on the availability of extensive standardized digital data to train the algorithms, which depend on the degree of the workflow automation, homogeneity, and reproducibility of the obtained data, the selected staining technique, the used antibodies, the selected genes, and hotspots of a testing panel. Hence, inter-lab validation studies are necessary for a successful application and transferability of machine learning models. Comprehensive guidelines have to be established to ensure standardized method outputs that can subsequently be fed into the machine learning methods. عندنا كمان ethical challenges خاصة بالpatient privacy and safety, fairness and inclusiveness, accessibility. عندنا workload challenge. It's to develop models that generalize well and are largely independent of workflow. The black box challenge, the deep learning still مش مفهوم قوي لكتير من الناس، فعلشان الناس تثق في النتائج بتاعته محتاج شوية وقت. Conclusion and take-home message. My number one take-home message: the AI is not here to replace hematopathologists, ولكن هو علشان يساعد ويزود الefficiency بتاعتنا ويقلل الوقت اللي احنا بنستخدمه في الروتين. Thank you so much.
شكرا للدكتورة. والآن دكتور أحمد البحيري البربري. أحمد البربري، اتفضل.